Welcome to solverfy.
So this is a long problem, so I'll throw in some basics first. So what this is going on about is there are different combinations of proteins that they are testing in this experiment. These exist as dimers, meaning that the T1R1 is a single protein, and we're saying it's bound to another single protein, and these come as a complex. So these are an example of the kind of dimers we're talking about. We'll say it's a T1R1 protein and it's bound to a T1R2. There's two, so it makes a single dimer. The response only happens because these two proteins are together and they do not exist in the membrane as a single protein eliciting a response. The other thing they're doing is they are knocking out the presence of these proteins in the mice. So what they're doing is they are finding the genes that produce these proteins. That's a 1. And they're making different kinds of mice that don't have these proteins. So what they're doing is they're knocking out, so they're preventing these proteins from being made, one of each of T1R1, T1R2, and T1R3, and they have a special fourth mouse where they knock out both T1R2, and T1R3. So in problem A, they're proposing two different models for how our taste receptors work. They're saying that there is a cell model and that there's a receptor-based model based on how our tastes work. The cell model is saying that we have taste cells, and each of these taste cells has one specific type of dimer. So we're saying this first cell has a T1R1 and we'll say a T1R2 receptor and no other kind of dimer. A different taste cell we'll say will have a T1R1 and a T1R3. These each have specific different types of cells, so we're saying that the tastes these different cells react to will be different and independent. The other kind of model they're proposing is that each of our taste cells have multiple sets of those dimers. So they're going to have each combination of these, and the signal it sends out is going to change based on which receptor or which dimer is activated. So it's going to have the whole mixture of these reactions. It's going to be the whole compilation of these different proteins in their dimer forms. So they're noting that in previous work, they took individual cells and they isolated them, and they probably ran a gel electrophoresis to see what kind of proteins they could find from each cell. So they ran multiple gels, and they found out that between different cells, taste cells, they had different proteins. So we're going to say this one did not have the T1R2, and this one did not have the T1R3. So since we're finding cells that do not have a mixture of all of the proteins, and they each only have certain types of proteins, the previous research they've done supports the cell theory, where each taste cell is different in their protein contents rather than containing all the dimer combinations at once. Problem B, they're testing the different types of taste. They're testing salt. They're testing bitter. They're tasting umami, which is kind of like the flavorfulness you find in meats. And they're tasting sweet. So they're testing them on knockout mice. You'll remember that the T2 T1, R2, T1, R3 is responsible for sweet, and the T1, R1, T1, R3 is responsible for umami. So in the knockout mice they've had, where they've knocked out, say, the combinations of these proteins, all of those knockout mice, even if they don't have these special combinations of dimers in them, they can still taste salt and bitter. This is kind of proposing that even without those special dimers, you're still able to perceive those different tastes of salt and bitter. This means that all our tastes aren't dependent on the dimers that were knocked out in this representation and experiment. In part C, they're showing us a graph of the response for an umami taste in knockout mice, where if you eliminate either T1R3 or if you knock out T1R1, you're not going to get any response or extra licking from an umami flavor. You'll also note that for the controls in the experiment, they used IMP to increase the flavor of umami, and they used a melanide to reduce the salty flavor. This is just to help control the licking the mice might do for the ingredient they use to prevent the mice from liking it due to a salty flavor. They're controlling it so that only the umami flavor is what the mice are going to lick for. So for this model, it's more representing that umami has to exist in the dimer form of T1R1, T1R3 to get any sense of umami flavor. If you knock out either of these proteins, you can't taste umami. So this is applicable to both cell models because regardless of whether the actual cell you're experimenting on has just the T1R1, T1R3 for umami, or has the mixture of all the different dimers, if you knock out either of these two proteins that make up the dimer, you're not going to get any response of umami from either of them. So you know that umami has to come in the dimer form, and regardless of whether the dimer exists in just one type of cell or exists in all type of taste cells, you're not going to get a response if you remove that dimer. Part E, we're given another model for a sweet taste. This one's a little bit different than the umami. Here you have the wild type or the T1R1. Wild type just means none of the proteins have been removed, so they have all the different types, T1R1, T1R2, and T1R3. So they're just normally born, not genetically modified. The other lines represent the removal of T1R2 only, T1R3 only, and both T1R2, T1R3 that make up the sweet flavor dimer. This model is more representing that if you remove any of these processes, T1R2 or T1R3, you're still not going to get as much of a sweet flavor. So again, you pretty much have to have the dimer to get that full sweet flavor taste. And if you knock out either one of those proteins, your ability to detect these flavors is greatly reduced. The only difference here is that the x-axis measures the concentration of the material, and at very high concentrations, you can perceive the flavor of sweet to a small extent as long as you have either the T1R2 or the T1R3 present. So even if for some reason you can't make the dimer for it, if you have T1R2, you can still perceive sweet, which is unexpected. That kind of is a lot different than the umami, where you must have both in order to perceive the taste. So moving on to part F, here we have perceiving the taste of sucrose as sweet. This was what I was referring to earlier. If you don't have both of the proteins T1R2 and T1R3, you can't perceive sweet at all, but it's unusual that if you have just a single of one of the proteins, it doesn't have to exist in the dimer form. It can somehow exist probably with a T1R2 and some other sort of protein that they're not testing in this experiment. Lastly, in part G, they're substituting kind of the flavors that are being perceived. They're using tetracycline, which is an antibiotic, so it's not going to have any kind of particular flavor. They're using it to substitute a sweet taste, and they're not going to use any kind of sweet ingredient instead. What they're doing is they're altering the protein so that when you apply tetracycline to the dimer, you're going to perceive it as a sweet flavor rather than no flavor at all. In this circumstance, it is kind of proving that you don't need specific substrates or substances to stimulate the dimer protein to get the response you'd expect. And it's saying that you can use other substances instead to trigger the response. That's kind of not what they're going for in the experiment, but it still manages to prove what the scientists are thinking, because as long as you stimulate the dimer, regardless of what you give to it, it's going to trigger a response that your brain perceives as flavor. So it's saying that although our dimers may not need a certain sweet flavor in order to send a signal, it's saying that you need a signal to perceive the taste of sweet. If there's no signal sent by the protein, you're not going to get that flavor being perceived.